I think we can start. Welcome to Gebze Technical University Bioengineering webinar series organized with Gebze Technical University Bioengineering Society. Today, our guest is Hasan Buğra Çoban from İzmir Biomedicine and Genome Institute. Let me introduce our guest today. He graduated from Ege University Bioengineering Department in 2006, and he went to University of Crete Biology Department, and then he completed his master's in Ege University Bioengineering Department in 2009, and then he went to the USA for his PhD and completed in 2014 in Pennsylvania State University, Agricultural and Biological Engineering Department. And he completed his postdoc studies in Washington State University in 2016. And he also had postdoc in Pacific Northwest National Laboratory. So you can start, Hasan Hocam.
Thank you so much, Batuhan, for the introduction. I'm quite flattered. Thank you. Good morning, everybody. The early noon time. Thank you so much for sharing this time with me, and I thank you. I thank Gebze Technical University Bioengineering Department for providing this opportunity to me. So, as Batuhan introduced me about my educational background, I will just skip this part. And today I would like to talk with you about general bioprocessing and how is it really involved in our life, and simply what I have done so far during my educational work about bioprocess. And again, if you don't understand anything in terms of like you cannot hear me or you cannot understand any technical explanation, please just let me know. Turn on your microphone and you are free to ask your questions. So, looking bioprocess word in linguistic, bio, we should have something living, the whole cell, organism, or something taken from the living organism, cell, tissue, or whatever. And process is the series of actions taken to achieve a particular goal. Before we start bioprocess, I would like to talk about our daily life physicochemical processes. The first one is very common for all of us, maybe brewing a tea. I'm not much a really tea person. I prefer coffee in my mind generally, but it's not really easy to brew a tea, a delicious tea for people, especially like you need some, again, processes to achieve a delicious tea. So there are some different ways, like you boil the water. Sometimes you just put the water first and you just put your tea leaf, or you put the tea leaf first, or you just add the boiling water. And some of my friends who are really expert on brewing tea, first they wash tea before they brew it with cold water. It's like a pre-treatment before the process to achieve a better end. So because they don't want any dusty or blurry color, they want a dark color without any blurry. So they wash tea before brew it. Even some of my friends in USA, they just bring their teapot from Turkey. So in other words, they are just carrying their bioreactors with them. So we just brew the tea. And if we just look at this photo, like visually, it seems like quite delicious, a dark color. Visually, we did some tests. When we drink it, sometimes we say tea is fresh or tea is not fresh. What makes us to say that? If it is kind of bitter, maybe it is like overbrewed. So there is no xylose or glucose, carbons, or sugars are all eliminated, and we have a bitter taste of tea. So just simply brewing tea is not a really process, an easy process. Another example, cooking rice. So in this case, I consider my wife is a really good processor. She's really good on this. Sometimes I just like watch her as an advisor in the lab. I assume our kitchen like a lab. First, she fries the rice like a pre-treatment in order to have not a sticky rice at the end. She uses olive oil if we are going to eat rice in our family members among us. But if our guests, for example, they are coming from Malatya, she is using butter instead of olive oil. So continuing about this cooking rice, so again consider this like a process to give it your consumers. So you need to know also your consumers, what do they want. Again, as I said, like if our guests are from İzmir, my mother is using olive oil. If they are coming from Malatya, she is using butter. And she also adds some lemon juice, and she says it makes the rice not sticky and also white. Of course, like she doesn't know the interaction between acidity and the solubility of the sugars, plant sugars. I know all the chemical and the physiochemical reactions while I'm cooking rice, but I cannot cook rice better than my mom because of experience. So in all of these processes that I had talked about and I am going to talk about, we can read theoretically, but we should have hands-on experience. It's the most important bullet. So, and when we switch on a real bioprocess, we can just simply talk about a yogurt making. So in this picture, you see a dew. It's just like accumulated in early mornings, you know. It collects all the microorganisms in just one single drop. I hope they seem, I wish they seemed like this under microscope, but I like this figure. So it's just like a mixture of lots of microorganisms in this single dew. Assuming it just like drop in this milk, actually like party is started like here because milk is a really high nutritious source for these microorganisms. But the single issue starts here that assuming this green microorganisms, they are lactobacillus, they have genetical background to secrete necessary enzymes to convert nutrients inside the milk to make them bioavailable for themselves, so they can rapidly increase their numbers inside this milk. But the rest, they are not available to use nutrients inside the milk. So they will eventually die, and the lactobacillus, they will increase their number, and they will produce their byproducts, for them lactic acid, but it eventually coagulates milk protein casein, and we will have agar yogurt. But thinking about everything, these microorganisms or lactobacillus or any organisms that we are using in bioprocesses, they don't really carry if the yogurt they make is good or not. They just want to survive like all of us under certain conditions that we created for them. So a little bit of production about production. Wine making is developed in Eastern Mediterranean before Christ, like 10,000 years ago. Of course, it's not the first need of human being to make wine, but it was just a simultaneous process. Maybe you are familiar with Cappadocia and the Ephesus Virgin Mary House. I worked as a guide for a while in Ephesus, and you know, in Aegean Park, we have lots of fig and grapes. And during summertime, it's so hot outside, and there are lots of consumers like birds or animals, everything. So people started collecting these grapes, which like has lots of sugar inside. They collected and store in the case. So outside is 35 or 40 Celsius in the summer, but inside the caves are almost like around 30 or 28, which is the best temperature the yeast likes. And you know this blurry color on the grapes, they're exactly their yeast on the surface. So they accumulated all the grapes in one side and the juice leaked. So basically it combined with the yeast, they had to wine. But it was the one of preliminary food storage condition. Then, second step, beer developed and cheese making, vinegar, and algae was cultivated. So they cultivated, not spontaneous anymore. So fermentation term is used. It's like around 16th, 17th century beginning of. And meanwhile, you know, like we are in 2020, and the CRISPR technique was awarded with a Nobel Prize recently. You know, we developed new technologies. So when we decide to do something using with cells, what we consider, like we can start from bacteria, the simplest structure so far, and we can go till animal and insect cells. So first of all, we need to know the product that we want to produce, especially molecular weight and how complex is about bonds inside the molecule and PTM, post-translational modifications. So we can secrete like lots of things in bacteria, but it's like a one single sentence. In order to make it meaningful, you should have some like punctuations. So some of them can do these post-translational modifications, but some of them not. And where we are going to use this product, what's our goal in like any therapeutic protein or like any vaccines or assays? Yield and productivity. How much like concentration be produced, and rather than yield, I really like the term productivity. How much concentration be produced in a manner of time? Because like I am sure you see in some articles, like for example, from different departments or institutes, they say that, okay, we produce like 50 gram per liter of, and previously the highest was stated in the literature 30 gram per liter. But maybe 30 gram per liter is produced in 1 week, but 50 gram per liter is produced in 1 month. So definitely I still prefer 30 gram per liter in 1 week. Productivity, because time means money in industry, and of course the cost. So An overall evaluation starting from a bacteria, E. coli, if you go to mammalian cell. Let's talk about the advantages of bacteria under the optimum conditions. E. coli, like repeating itself every 30 minutes. This is great. So your workers in the bioreactors, they are just multiplied in every 30 minutes. It's great. But comparing to mammalian cells, if everything is going well, you need at least 24 hours, or maybe at least I've seen like 18 hours, but mostly even it's more, sometimes 2 days. And complexity of growth medium, you can really like grow E. coli in a simple medium chemical composition. So it's really with a low cost and expression level, you can really, and some genetic modification, you can express in really high levels. Starting with the problems. Secretion to periplasm. Really a few number of articles stated in the literature that, or we need to do some modifications to make E. coli to secrete extracellularly based on the product. And as I mentioned previously, post-translational modifications. E. coli, unfortunately, it's not really successful to make post-translational modifications. But if we look at mammalian cells, okay, we stated some disadvantages like they grow slow, they need complex grow medium, but they are able to make all these complex post-translational modifications. And as you know, like about the current topic, coronavirus vaccine is trying to produce using mammalian cells. Again, we need to know what we are going to produce, what the consumer really wants, and how much we can spend. Because, as a simple example, if you need to buy a bottle of water, you can just go to grocery, like you can try E. coli. But if you are going to buy, let's say, dragon fruit, then okay, you need to go to a shopping center, mammalian cell. So again, we need to define our needs. So briefly, I will talk about my studies starting from PhD and the postdoctorate and the current studies briefly. So during my PhD, I worked on 4 different projects. One is production of phenylpyruvic acids, which is the deaminated form of phenylalanine and has almost the same nutritional level, but used for kidney patients to reduce urea accumulation in their body. Second one, production of phytase enzyme with Aspergillus picum. And the third and fourth one, they are about food safety processes. The third one is in a milk collection system, how to sterilize the CIP and the setting up a CIP system. And the fourth one, the contamination of cheese slices. So I will skip phenylpyruvic acid project for the sake of time, but I will talk about some phytase project. So phytase, I'm sure many of you or all of you, you have already know, is an enzyme which breaks down phytate. And the phytate is a compound that's abundant in the legumes. And people, like for example, in Sri Lanka, who consumes legumes in their diets, they are having some health problems because of phytate, which has like 6 minus phosphate group in the structure. So because of this minus groups, phytate creates challenge with divalent ions, for example, calcium, iron, and it causes iron deficiency or tooth decays or weak bones. So our goal was to produce this phytase microbial way to break down phytate like a pre-treated food and to serve it for first animal consumption and then human consumption. So based on the literature search, I come up with the 4 microorganisms, and just without any optimization in a simple shake flask production, I can see that Aspergillus ficum showed a really and significant difference in terms of titer and the unit per milliliter enzyme activity comparing to rest, and we want to go our further studies with this microorganism. So I use mini-type optimization condition response surface to optimize first growth conditions, and I focus on temperature, pH, and aeration in the 2-liter Sartorius bioreactors B+. So I come up with 15 program algorithm, come up with the 15 runs and 3 with replications. I run the production under these conditions, and I measure the enzyme activity and glucose residual. And based on the model, this measured and predicted, we created a model I will talk about later. And the algorithms say that if I ran my system under 33 Celsius and the mild acidic conditions, almost 4.5 pH and almost 0.9 VVM conditions, we can have 2.28 unit per milliliter activity, and we confirmed it with 3 replications in the bioreactors. And I did the same optimization steps for medium optimization and then fed batch continuous modifications, and we increased the phytase activity, especially in the fed batch productions, and productivity in continuous fermentation. But however, I'm sure you have, I have some attendees here, they have already worked with fungi before in bioreactors. By the time they create a bulk growth, as you see in this bioreactor, they just bind to each other, especially around the shell and the agitation system. So you say that, okay, I have a really big amount of biomass, so you assume that you will have a good yield at the end of your production, but you may not, because in this structure, just outside of this biomass is connecting to the environment in the inner environment of the bioreactor, and inside there is a strict biomass transfer problem. And the inside is especially lack of oxygen, so most probably, or for sure, they are switching their metabolism to something else that you don't want them to produce. And they are just consuming your food you provided for them for something else. So you're just wasting time and the money at the same time. So we come up with the idea, and that we saw in the literature, to use some microparticles. So those microparticles, they can be talcum, which is like a magnesium silicate, and the baby powder, which is basically, or aluminum oxide, to break down this bulky growth and have smaller structures of pellets. So just addition of talcum and aluminum oxide in shake flasks, previously we used to produce 1 unit per milliliter, but just adding talcum, about like 15 grams per liter, we increased our yield by 3 times. And we implemented this in the bioreactors. This is image is taken from the fungi pellet taken in the 17th hour of production, I think, because after 17 hours, they really just stick to each other. From B to F, I add different concentrations of aluminum oxide, and you can see this like black dots there. Aluminum oxide is also inside the pellet structure, so it also allows mass transfer efficiency inside the pellets. From G to K, it includes like different concentrations of talcum. Talcum is a finer molecule compared to aluminum oxide, so it's not really easy to see in this photo. But just using this, we change the fungal structure and the increased mass transfer and the productivity. So again, with some optimizations, I increased further phytase concentration to almost 10 units per milliliter. So we started with the shake class almost 1, and with the optimizations and engineering approach, we increased till 10 units per milliliter. So another project I have done in Penn State, it was a TIP cleaning in place system of a milking system using electrolyzed oxidizing water. We used Mr. Farm. So how was the system? Milk is collected from the cows and it just goes through from a pipe system and collected in a collection unit. So after each milking system, this tubing and all connections, gaskets, and the collection tank must be cleaned because it's quite hot because of animals over there. Even winter time, it is still warm. So it's really appropriate and a great environment for microorganisms to grow in the leftover milk in collection system. So Mr. Peachy, he used to have a conventional system. So basically he used some like acid and base. He just purchased from outside and he used some hot water and pump it through the system and wash it out. We came up with a better idea based on our hypothesis. And after cleaning, we did some testing like microbial presence, which is like, yes, there is contamination, microorganism is growing or not. Or we use ATP swapping. Maybe some of you are familiar with this. When you swap your sample, there's an enzyme, luciferase. So it's just like a lighting box at night. They have the same enzyme. If there is something like living, it means that they produce ATP. So when it connects with the enzyme, it just gives you a light. An amount of light is converted to a number of microorganisms with this one single ATP swapping. So in our method, what we have done, we basically used a sodium chloride, which is a basic salt, and using an electrolyzed system, oxidation system, we created an acidic water and alkaline water with here. So what we have used is cheap salt and some electricity. So in a conventional CIP, Mr. Peachy, he used some rinse, warm water, then alkali wash, then acidic wash, and that's it. In our system, we use some warm water to wash out the leftover milk first, then we use alkali wash and again rinse water and acidic wash. So I took sample, yes, this is my hand, from like mid holes, linear elbows, especially like in terms of fluid mechanism, the most problematic points, and the gaskets and straight pipeline, which is like fine. We don't have any like big problem about straight pipeline system. So based on FDA regulations, we made a comparison between our system and the conventional system, and in all sampling points, our system was more efficient to conventional system. Only we had like couldn't achieve to blow FDA regulations in H point, which was an elbow. So in that case, we come up with the solution. We need to change and find another connection rather than a sharp elbow. And about an economical comparison, each CIP cycle cost $2.15 in our system, and for conventional system, Mr. Peachy spent $2.84 per CIP. Am I going fast, or do you have any questions? Please, meanwhile, let me know. Because rather than like a lecture, I would like it to be like a communication. So my fourth project in my PhD, we had a pulse UV system to be used in some decontamination processes. So you see an equipment on the right lower corner is our system. So you know we have like a UV light in our laminar flow cabinets, and it's a constantly supplying UV light. But in this system, it's just like store power and punch once. Why do we need this system? Because like sometimes there are some products you cannot really use heat to sterilize. For example, lettuce or strawberries. And there is no outside peel, like you just need to wash them and consume. But sometimes washing is not enough for food safety. So what does this machine do for us? When it like send the UV light punching once, it breaks down adenine and thymine connection, and basically pathogens, they cannot replicate themselves. I just want to like clarify this graphs for you. We put our sample in Petri dishes, and we put like an amount for 12 milliliter, 24 milliliter, and 48 milliliter. When we put 12 milliliter in just like one single shot, we achieved almost 8 log reduction in the pathogen concentration. If we have 14, we need to wait another second. If we put 48 milliliter, we had to wait for 5 seconds, and that's it. Not more than 5 seconds. So each this data line 1, 2, 3, 4, and 5 every second, in 5 shots and in 5 seconds, we are totally clean. And in this, under this 5 seconds, the temperature of our sample will increase, still under same as room temperature, but under further shots, temperature increased almost to 50 Celsius, which is not really wanted for our sample. So, looking at the figures on the left, they are like a Staphylococcus aureus before treatment, so they are like entire and keep their integrity. After treatment on the right, some of them they even lose their integrity. They explode. Even you see still like some entire cells, genetical integrity, so they cannot replicate themselves anymore. So the process was like we take this sliced cheese and we contaminate it with penicillium and listeria, and we did some stomacher homogenization, and then after treatment, and the CFU calculations. We came up with some results that UV light is effective on both of the species, but less effective on fungi. So it's because of the formation of the structure or something more, or chemical structure of the microorganism. So briefly, I would like to also talk about some of my postdoctoral associate studies. In Washington State and the Pacific Northwest, people mostly focus on lignin degradation and the production of hydrocarbons and jet fuels and lipids, eventually to be converted into biofuel. So talking about the first study, a little bit slow. Okay. So you know, cellulose and the lignin. Lignin is also a really great date to be used in microbial productions. If you remove like glucose, all the carbons and cellulosic biomass, lignin leftovers still can be used with some microorganisms and can be converted to lipids and eventually for fuel or alcohol. So lignin is the second most abundant biological material after cellulose. So it's everywhere. This is great. And we can use it for a renewable source production, second generation biofuels, not first generation. But it is highly variable, even from the same tree or let's say like plant source, based on the season, based on the geography, the composition of lignin varies a lot. And by the degradation of lignin, there are lots of phenolic compounds for microorganisms. They are poisons, really inhibitory. So it's not really easy to grow microorganisms just by using lignin, but we are trying. So actually, unfortunately, I'm not really allowed to share all these details because the project is still going on over there. But we tried to grow 3 different microorganisms. They're all Rhizococcus strains. And the Rhizococcus opacus PD630 really growing well. So it is great. And we created a genetically modified version, and it's not really growing well, but still it was a good worker for us. Because on the right side of this graph, you see co-fermentation of PD630 and vanillin A. Biologically, break down the lignin at the most state with this co-fermentation, because Rhizococcus vanillin A minus degraded the lignin till to create a vanillin structure. And that vanillin is further used by PD630. And we did some analysis about lipid composition, and the 14 carbon structure was the most. And we used the red dye to show lipid inside our cells. And the second of my project, I work in the postdoc. In this case, again, we used lignin and the plant source to be catalyzed by lactase enzyme provided by Tremates versicolor, which is a fungi, again to create some hydrocarbons. And we did some GCMS analysis. Okay, so it's shown, and it includes, just like in the microbial environment, and after, of course, extraction process, we have undecane and dodecane, which can be like tetradecane, can be used as biofuels. And my last project in postdoc, I was using a 30-liter, again, Sartorius bioreactor. I put this figure, and this was again a lipid production for, eventually to be used as a biofuel. It was a really like real-time automated system. I really like this. I set up everything, and I was able to control it on my iPhone at home. And maybe you see a probe like this rectangular structure at the bottom of the bioreactor. It's also purchased from Sartorius. It was like almost, I don't know, I'm not sure, but $50,000. It was a great probe. It takes like sample every second and measures your microbial growth and carbon consumption and product formation and everything, and come up with the graph. Like, you just inoculate your bioreactor in the evening, just go home and sleep. You come back to lab in the morning and your graph is ready. So, and recently, what I'm doing so far in 9 Eylül University, and when I was working in IBG Center, we mainly focus on like rare disease or some cancers to produce some antibodies or antibody fragments. Biosimilar production was really highly worked in IBG Center. So biosimilars, as an advantage, they are not chemical, they are not synthetic chemicals. They are still like produced in biological systems, and they are really highly specific. So and biosimilars that we need to partially mimic a product. So we have a reference product and we analyze it, the structure and everything, how it functions. Then maybe it's better if we move on this step. We create a vector to make the cell secrete this, and again, with the bioprocess system, produce in a bioreactor and purify it, and the final product serve it. So specifically, I use the E. coli to produce an antibody fragment. Please be careful about here the term antibody fragment, because at the beginning of my presentation, I mentioned that I can produce the whole, that Y-shaped antibody in mammalian cell, because it's a really big structure. I cannot produce it in E. coli. But when I look at this Y shape of antibody, I know interactions just go on this paratope, epitope interactions. Just like think about my nails, all on the structure. I just need this much. Okay, the molecular size of this fragments, let's say like a CFE structure for the antibody, if I just produce this, I can still use for patients, vaccine, or for to treat something, a real rare disease. Okay, so I just create with the synthetic plasmid design of this structure just to produce these two nails in my finger. So in that case, I can produce it in E. coli, what I have did in IBG Center. Of course, what is the disadvantage? This is Y shape. I don't have this tail anymore. So this structure, small structures, they are not stable as before structure, but still it is much way cheaper. So what I have done, Of course, my friends, they designed the plasmid, and they gave me the plasmid. They said, okay, Buğra, this is the plasmid and this is the cell. I made the fusion and start the production. And in this case, you know, when we say E. coli, we commonly think about 37 Celsius. But my goal is not to produce E. coli, of course, to keep it comfortable for a while. But my major goal is to produce the product that I want to produce. And when I produce under 37 condition temperature, our produced protein start to coagulate. And we had lower amount and function of protein in our production. So in this case, we decrease the production temperature to 30, then 18, and even 4 Celsius. So in 4 Celsius, of course, like we used to finish our production maybe in 14 hours. We wait a couple of more hours for E. coli to grow and we utilize the nutrients, but we increase our production more and more. And after production, how we collect our proteins during plasmid design, my friends, they use a special tag at the end, so only our protein of interest has this special tag. So in an affinity chromatography, just binds to that tag, and I cannot give, unfortunately, all these details because it's still going on project. It's just like an affinity binding, and we can, with this simple and 1 or 2 step purification, we have highly purified target of protein in our hands. So yeah, we just came to the end. These are my publications. If you are interested, like some of my works, I would be more than happy to talk about. And in my future plans, academic plans, I would like to still continue work about microbial-based studies, and I really like to use what in defined as waste. I want to come up with something valuable using a waste, and green energy processes, some value-added products and recombinant proteins. And when I use a bioreactor, even it's a purchased one, I still like to modify the bioreactors based on my approach. And currently I was using one here to increase oxygen solubility. And some courses I am currently and in the future I can teach about introduction bioengineering, bioprocessing, and bioreactors, and some upstream and downstream processes. And the seminars that I really like to teach to my students here, because I also get some body language education, when I maybe show a pathway for them, what they should or shouldn't do during a presentation, oral presentation, or poster presentation, about mimics, gesture, posture, and using their voice. So thank you so much. Please, let me know if you have any questions, suggestions, or comments, please.
Thank you very much for your informative presentation. If you have any question, you can click to raise your hand button. Yes, Feroz Albana, yes.
Yes, first of all, hello everyone. I'm Feroz, the Vice President of GTU BIOS, Bioengineering Society. And on behalf of BIOS, I would really thank you, Dr. Hasan, for this great presentation. It was really full of great information. Also, we have the pleasure to learn from your previous experiences and experiments. Moreover, I would like to thank our guests who joined us today. Plus, do not forget to check our websites for our upcoming events. And if you didn't check our first magazine issue yet, please have a look at it through the link in our Instagram page. And last but not least, we're really pleased that you shared your vulnerable and experience with us today, Dr. Hasan. And thanks for everything. We're looking forward to know more and learn from your future plans. And we're happy if you shared them with us in the future. Thank you, everyone.
Thank you.
Any other questions?
And also, I have already left my email and even phone number. Please you can email me or call me whenever you want about studies or any like potential partnerships. I've been happy to talk about it.
Yeah, seems like we have no questions anymore.
Okay.
Thank you very much for coming and presenting in our webinar series.
It's my pleasure. Thank you so much for your invitation.
Thank you, and see you in the next webinars.
Sure. Bye.
Goodbye.